Are you connected with everything?
I think I am connected, yes.
Thank
you. Thank you, Ola. So I'm here to talk about the clinical sequencing facility that we have recently started operating here in Uppsala, which is also part of the SciLifeLab. I can just mention that there is another clinical sequencing facility in Stockholm that also are running clinical sequencing, and they're called Clinical Genomics. They've set up their operations in a slightly different way, but if you want to know more about them, you can talk to us later or visit the webpage. So one overall conclusion when you listen to the talks today is that research or next generation sequencing in the research setting has come a very long way, and there's lots of research infrastructure for next generation sequencing. However, when we are looking at what we are doing in the clinic or for the clinic, we have not really come that far. So we're not really up to running these huge numbers of sequences in parallel, and we're not really interested in processing that much data yet. But of course, things will likely change in the future. So I think I will start to take a step back and introduce some of the reasons why we are interested in setting up next generation sequencing in the clinic or for the clinic. And then I will go through some of the challenges that we face as we want to do this and present some of the work that we are doing now today. So obviously, whenever we are doing translational science or something, we're saying that we're doing something for the common good of the patient. We want to improve the diagnosis. We want to get a better disease subclassification to make things easier and better for the patient. So hopefully, if we are able to subclassify disease better, a physician will hopefully be able to put a better diagnosis to a patient quicker. For instance, in certain cases, it might be that today they will receive a standard treatment for a disease, and that drug might not function very well in the first time. And then the patient doesn't really respond to the treatment, and the patient will receive a second drug or even a third drug before it actually responds to the treatment that can be given to the patient. With a better diagnosis, hopefully the physician will be able to give the correct drug at the first attempt, and we will hopefully be able to reduce the time it takes to get good treatment, but also have less side effects, etc. for the patient. So all these things are for the common good of the patient. In cancer, we will also be able to monitor the disease progression in another way than we do today, and hopefully we can detect when we have a tumor that reappears in leukemias, for instance, or elsewhere, earlier than we do today. So all these things are very good. But there are also more economical or other issues that are good with next generation sequencing. So today, with the Sanger technology that is used by most of the hospital labs today, whenever we are looking at several different genes at the same time to be able to diagnose a patient, we will have to run one assay for each and every target that we need to look at. And the more targets that we're looking at the same time, the more expensive that analysis will be. While in the next generation sequencing setting, we can have a gene panel or something similar that can just, when we run just one assay, and we will have the same cost whenever we're running this. So in the long run, this means that we will probably end up having cheaper assays in the clinic. We're also aiming at better sensitivity, because with the Sanger technology, we have the read peak is really the mean, the result of the mean sequences that we have in the test tube. While when you're running the next generation sequencing experiments, obviously each read is going to produce, each molecule is going to produce one read, which means that we can also have higher sensitivity and detect low frequency variants in an easier way. So apart from this, we can also argue that we will have less pipelines running in the labs. Hopefully we can have, for instance, one exome pipeline running most of the inherited disease panels in the future, and then we can have in silico panels, for instance, to look at at different panels for different diseases. So these are all motivations why we want to take this to the clinic. So, but we need to think about certain things as we do this. So obviously, the sequence information that we get from the next generation sequencing instruments needs to be validated in the clinical settings. We don't want to end up in a situation where we have poor analysis that we use for the patients. So also, we are working with the clinics that have accredited tests that they have for the referring physicians. And we need to have accredited tests also for the next generation sequencing test that we produce. And as previous speakers already mentioned, the secure handling of the next generation sequencing data is an absolute requirement. And in the hospital setting, of course, that cannot be compromised. And today there are not that many guidelines when it comes to how next generation sequencing should be reported in the clinic. And these guidelines have to be developed and agreed upon by national work groups. So if we try to sort of split the different challenges that we have into different parts, obviously what people talk about a lot today is the infrastructure. Oops. Sorry about that. Is the infrastructure part of it. And we have just recently started our operations and we have begun to think about this and have implemented a few of the things. But what we really need to do is to design really efficient and secure data flows, both within and between the university and the hospital. Something that is relevant for us is that the IT system in the hospital is supposed to be completely closed. So we need to find good ways how to transfer data from within the hospital to the university setting and the calculation capacities here in Uppmax and also the other facilities that we have. We also obviously need to implement pipelines or workflows that are good for the clinic. We need to, together with the clinicians and the geneticists, we need to interpret and report on the different variants that we find. So that links to the diagnostic challenge. Whenever we are using next generation sequencing, we're probably likely to find a lot of variants that are not reported before as having a clinical impact. And we need to define rules on exactly how to act upon variants that haven't been previously reported to be clinically implicated in disease. There is also other thoughts to think about. I mean, we're a team of people working with this, including clinicians, geneticists, and bioinformaticians, and we might have different objectives. We might have different ideas about how solutions how they should be, and obviously that has to be coordinated as well so that we can agree upon what we produce. We also quite often have sample inputs that are not easily worked with. The pathology department, for instance, they want all of the analysis pipelines that we produce to work on formalin-embedded tissues because that is what they have. We might have swaps in the future or other kinds of materials. So obviously that is a challenge for the lab personnel in particular. And then we are also very much concerned about the legal and the ethical issues, as previous talks have also mentioned. So if we compare this to the research setting, there are a few differences that I think I wanted to mention. So obviously, processing a large number of genomes is not at all as important as the speed to process a single genome. For instance, we don't really want a kid with leukemia to wait for a diagnostic response. We want that response to get back to the physicians as soon as possible. And that is also why the infrastructure downtime is not really something we want to accept. A second thing is with the accredited tests. It might be that whenever a software is updated, which happens quite frequently in this community, it might have to be validated that it actually corresponds to analysis that we have done before. Any database updates when it comes to annotating variants, for instance, might also be an issue. And there might be chemistry updates also from the vendors. So all these might require validation, and it's something that we need to control. Also in the research setting, whenever you find a variant which is genome-wide significant or something like that, and might result in a science publication, you're really excited about it. In the clinical study, that's completely unuseful. Unless you have very good reasons to believe that that variant has a really strong effect on the disease that you're interested in. You're really interested in finding what is already known. And you need to report on known variants that have an implication in disease and say that we really sequence this variant and the patient does or does not have that particular variant. And we need to know that we have enough coverage on that particular variant, but the patient didn't have it. So the novel variants are rarely actionable clinically, and we need to report on what was not sequenced. And then we also need to think about that in Sweden, at least, I don't know about other countries, quite often the clinics don't consent people on a regular basis. So it's something that we need to think about when we send the genomic data to other servers or store it in databases, that it is really not consented data always. So it's important to keep the non-consented data and clinical information and so on inside the hospital. And the hospital IT system is perhaps not as developed for next generation sequencing data as the university IT. Something that I didn't mention on this slide is also that some of the tools that are very common in the research setting, such as the GATK, requires a commercial license when you're working in the hospitals. So, for instance, if we are interested in including GATK in our pipelines, we would have to pay something like 250 or 500,000 crowns per person and year to be able to do that, which would sort of make everything a lot more expensive for the hospitals. So how do we go about to set this up in the clinical practice? This is a schematic of how the clinical sequencing facility has been set up here in Uppsala. So what we do is that we have split our work into work packages that are focused on different kinds of diseases. And this follows the structure of the hospital in a way. So in work package 1, we're working with the pathology department on solid tumors, and in leukemia we're working also with pathologists and with the clinical geneticists. And with inherited diseases in work package 3, we're working together with the clinical genetics department. This means that we have about half of the staff that is linked to the clinical genetics department, the clinical sequencing facility, is also employed or working at the hospital. So all the lab work is being done at the hospital. We have put our sequences inside the hospital and they are run by hospital staff. And then the bioinformatics people are employed by the facility, working in close collaboration together with the different work packages, the other work packages. We're also setting up this for clinical immunology, but that's really something that we are planning to do. So we're trying to invent as few wheels as possible when it comes to these things. So we will try to use the LIMS systems that are already operating in the clinical departments, so that the lab personnel who are already used to working in a specific way at the hospital doesn't really need to learn again how to document and work with the lab flows, which means that we can also use the accredited systems that they already have. And then when the data comes off the sequencers at the clinic, then the bioinformatics starts, and we have all the standard steps that we go through, all the QC and the alignment, etc., all the way. And we have split our different projects into two types of projects. So we have one pipeline that we would call diagnostic pipeline, where we have different gene panels that we have already validated and are running in the clinical setting, which means that we would have, for instance, 8 colon cancer patients being sequenced every week, etc. So that panel is continuously running for the clinic. And then we have a second pipeline, which is really development pipeline and also research pipeline. So whenever we have projects that we accept to the facility, then we would initiate the research pipeline to sort of define all the analysis tool flows, the analysis pipeline, and so on. And then with the aim of lifting a research pipeline to become a production pipeline. What is also specific, a bit peculiar about our facility is that because we incorporate so tightly our work with the clinic, we're also collaborating with the geneticist and the clinicians at the hospital site, and we will follow the work all the way down to actually reporting back the variants to the patients. So we are involved in all those steps on that process. And then we plan to store variants that we report, and we plan to store our data in different databases, of course, as well. So at our disposal, we have 2 MySecs that are, one is at the clinical pathology department and the other one in the clinical genetics department. We also have a HiSec, which is right now situated in the SNP platform here in the PMC. We have a small server that we have for transferring data to back and forth, but also for some calculations and for temporary storage. It's good enough to process the panels that we are running, but whenever we get data from the HiSec, it's going to be a little bit more, perhaps not enough. We also obviously have access to the production cluster at the UPMAX, where we do most of our development work. And we have storage both here in Uppsala University, which is expandable. And we aim to set up more storage at the hospital side as well, where we also wish to construct different kinds of clinical databases. And we have a virtual server for different kinds of web applications. So something that is also very important for us now is that we need to set up national guidelines on how to work with next generation sequencing for each of the disciplines where we are active. So there are a bunch of different things that are still open topics, and there might be guidelines from other countries that could be implemented in Sweden, but for the clinic it's important to agree upon how to do this. And obviously we have initiated work groups with people from all over Sweden, from the clinical, from the different university hospital towns, for inherited diseases, with people both from the clinical genetics department but also other relevant labs. There is a national work group for pathology, hematology, and also there is going to be one for clinical bioinformatics. But we have also initiated work groups that are to work on how to deal with data security and data sharing in the clinical setting, and also about how to think on ethics and consenting and these things, which I think is of interest also for the research community at large. And then I just wanted to mention what kinds of panels we have running so far. So most of the ones that have been implemented and are running already are Haloplex panels. So we're running colon cancer at the pathology department, so it runs on formalin-embedded tissue. And then there are 3 different panels running on the clinical genetics department. We have several different pipelines, several different panels under development. We're also investigating if it's possible to detect structural variants in a nice way based on exome data, exome sequencing data. There is another Haloplex panel being developed for lung cancer, and then we're together with Adam and people at the Uppsala Genome Center, we're working on implementations with the PacBio data as well. But obviously we have lots of infrastructure work that we need to sort of continue working on. And so the data transfer between university and hospital is entirely manual today, so we need to get that automated. And the same is true for the transfer between us and the other SciLifeLab platforms. We're involved, we're supposed to be the national node for the Human Genome Project, which was recently sort of agreed upon. So we need to set up a database for that as well. And we need to set up databases at the hospital side as well. And there are also all these standard things about setting up project management and ticketing systems and so on to be able to accept projects in a clear way, as we're supposed to be a national platform. So that was it. Thank you.
Thank you very much. Yes.
How would you envision data flow from the databases with the clinic information, which is sort of hidden behind a lot of firewalls in the hospital, and the databases on the sequences and the results from the sequence data? Because these two things have to come together.
Yes, I think that what we aim for is having the variants that we report back to a physician also inside the clinical database, inside the hospital. And the question is whether if we should allow anyone access at all from the research setting to the clinical database, because that's going to be a clinical database only for the hospital, so that a physician can go back and query, in cancer, for instance, when we see this variant